only for the invitation, but for putting together, by far, the best school I've ever been. I mean, it's a totally unbiased opinion. Actually, when I was preparing this presentation, I was going back to take some pictures, not to recycle any slide. And I realized that today, 1 year ago, I was giving kind of the same presentation, not in content, but in the context here. I was a little bit more nervous for some reasons. But it's nice to, 1 year after, recap a little bit. So actually, what I'm going to present is my postdoc work. So it's work that has been going, or was going on, for about 6 years in the US, in Princeton, where I did my postdoc. And it's a collaboration with a bunch of people that I will start mentioning as they appear. But also because I've just started here. You see, I've been the whole week helping people getting this work. Yeah. Where? No. Yeah, I think it's because of the adapter. It's fine. I have a lot of animations, but I can use it. It's fine. Well, anyway, also because, as I was saying, I started a few months ago a job here. I also want to use this presentation to give you a sense of what's going on, or what will be going on here in terms of theoretical ecology. And the reason why I'm doing that is because a few weeks after I came back from this presentation I was mentioning last year, I was very happy because I was going to start a job here. And I was talking to some people in my department, and we were celebrating that I was going to have a job here with some uses. And at the same time, I was very nervous because I was like doing this big transition, feeling responsibility. And then I asked one of the professors in my department, how do you get ready to do this? And he told me that I would only need to learn one thing, but I would need to learn it to do it very, very well. And it was to find people that is smarter than I. And that's what I'm trying to do. It's not very hard, but I want to give you a sense of all the things that can be done in here. So if you're interested, we can talk. And if you want at some point to come here, it would be great. So switching a little bit more serious and starting into the topic, the first statement that I want to make, that is actually what the whole work we do here turns around, is that population dynamics is an emergent phenomenon. And the reason why I do that, I mean, we've been the whole week attending to super interesting and amazing lectures about population dynamics in different contexts. And I want to emphasize that all the work we do is considering that this is an emergent phenomenon. It's a macroscopic pattern that we observe. And it actually relies on some microscopic processes. So, for instance, if we consider one population, some bacteria that are in a plate all over the place, they are very well mixed and they interact with each other, then we have this kind of pattern for the dynamics of the population, right? The logistic growth that we all already know at this point. But actually, this relies on some microscopic processes that are happening at the level of the individuals that in this case are very simple, are reproduction of cells. So one cell divides into two. It's death, so one cell dies and disappears. And it's also some sort of competition. So at some point, the environment gets super crowded. Cells fight with each other for resources, for space, and then one of the cells is eliminated. And these processes result on this macroscopic pattern. One important thing is that these processes are stochastic. All these processes happen at some rate, and what we observe in this pattern is some kind of an average that is better the larger the population is. And of course, what allows us to go from this microscopic description to that macroscopic description is some sort of mathematical tools that at the end result on this logistic growth equation that maybe rewritten in terms of current capacity is more familiar to you all. On another example, if we consider two populations, let's say red and blue bacteria, one eats on the other, we also observe sometimes these macroscopic patterns, macroscopic in terms of time, right? Coarse-grained patterns, if you will. But they actually also underline on a different type of microscopic stochastic dynamics. In this case, we have different processes. In the classical Lotka-Volterra model, we have preys, in this case the blue cells, that divide at a given rate, predators that die in the absence of nutrients. In the absence of prey, they die at a given rate as well. And then we have the predation process by which a prey encounters a predator and the predator reproduces. And using the same techniques, we can go from the microscopic to the macroscopic process, and then we have the mean field Lotka-Volterra equations for a prey-predator system that we all know. So this is, of course, super useful in some limits. It allows us to do a lot of analytical work, to work with ODEs, which physicists will say are super simple. Biologists think are super useful. And we have all these discussions and actually very important and insightful biological results in this using these assumptions and working on this framework. But the main question that we do, or the question that I really enjoy making when looking into these population dynamic processes and into ecological problems more generally, is what happens when these microscopic processes that underlie the macroscopic patterns are indeed more complex than this basic death reproduction event. Indeed, there is way more ecological systems in which these assumptions fail than the number of processes that we have in which these assumptions are actually more accurate. So for instance, going back to this one population case we were discussing before, when we have only one population of blue cells, we have this logistic growth. And as I was saying in the beginning, we're assuming that cells are all over the place. They behave as an ideal gas for physicists. Every cell has the same probability of interacting with every other cell in the plate, has the same probability to interact with each other. But when we go into the field, into the real world, if you want, we find many systems. In this case, I'm showing you tracking data of 6 wolves in Minnesota, in which, as you can see, this assumption completely breaks down. I mean, it's absolutely wrong. These animals have home ranges, they are territorial, they don't overlap perfectly with each other, and the probability for the red guy to interact with the blue one is way, way lower than the probability of the red one interacting with the yellow one. And this is something that we can work out microscopically. We can work with random walk models to see the encounter probabilities. But how that scale to the level of the population is something that we are actually now trying to work out. So in this logistic growth, because of the assumption of the well-mixed hypothesis, at the end, the interaction terms result on a quadratic term, because it's basically related to the number of pairs that we can make in the population. But here, this assumption is totally wrong. So we expect more complex terms in this equation to arise, and probably more complex processes and macroscopic processes to occur here. So whether determining the limit in which this well-mixed assumption works versus the territoriality, of course, will depend on the species, will depend on the system, and will depend on many, many different variables that one needs to consider carefully. One more example that I want you to pay attention to, is actually relying on one of the other assumptions that these well-mixed models consider, and it is that encounters between individuals only happen when the trajectories of the two particles cross with each other, and they are totally random. So these guys are moving in straight lines until they find one of the walls of the recipient, if you want, and then they turn back, right? And that hypothesis is also super strong for many systems. We know that animals not move completely random. We know that they have perceptual ranges. They are able to see each other, and they are able to tune their movement depending on how they sense both the environment and their conspecifics and non-conspecifics, other animals, basically. So then the question is how this cognition, how this searching ability, and how these perceptual ranges may alter population dynamics processes. So something that we did way back in the time, well, way back, a little bit back in the time, and not that only in my PhD, was to consider that in models of animal searching to see how different groups of animals form. So here what you are seeing is an actual satellite image of the Mongolian steppe. The Mongolian steppe is a super flat region where vegetation is has a super random dynamics, appear and disappears very randomly. And the red actually represents better grass for the animals, and the green worse grass. It's an exercise of focusing really, paying attention to the colors. And the size of the star represents the size of the group of animals that, according to our model, forms in regions with more grass. So as you can see, nothing super surprising. There are groups that are bigger that form in regions that have higher quality of forage for these guys to feed. But of course, if my distribution of individuals is not homogeneous, again, I don't expect my interactions to be following this ideal gas hypothesis, and different phenomena may occur on different regimes. So at this point, I want to stop a little bit just to very briefly give a take-home message. These population level models that assume well-mixed hypotheses and everything, I'm not saying that they are not useful or that we should leave them. They are super insightful and they are great, but there are some regimes in which they don't work. And the questions that we follow is, what happens in these regimes where these models don't follow, and whether we can work more elaborated ones. That's the general idea. And an example that I find super interesting, because I felt like super interested the first time I saw these biological processes in action, is actually collective behaviors. So what happens when this population shows complex collective behaviors, and how those collective behaviors scale up to population level processes. Just to give you an idea of how these collective behaviors work, here we have some fishes that are actually they are feeding on the ground, and they are somehow going from top to bottom, trying to both keep the size of the group and the cohesion, and also make the foraging on reasonable times. So they are somehow circling back from the top to the bottom to feed correctly, or to feed fine. And then we have another example, which is the starling, the flock of birds that Bill was mentioning in his first lecture. I'm going to show some video where you can see how they coordinate with each other, and one of them turns, the other one does as well, and they form these like super amazing structures, and they have propagation of density. I mean, I don't know. To me, this is like a fascinating example. But I guess some of you are thinking like, okay, these processes happen like in hours, or they take hours. Community level processes and population level processes happen on way larger time scales. So how is it possible that this coordination of individuals scales up to actually having important and have a real impact at the level of the population? Well, in the case of these examples, it may happen because birds actually, by forming these flocks, they migrate more efficiently, and they might have some implications. But there are other examples in which this becomes a little bit more clear. So here we have the formation of swarms of locusts. Basically, locusts are insects that they live. They are solitary. They don't have any kind of collective or social behavior. But once they are surrounded by many other locusts, they form these swarms that allows them to move a lot, and they become more social, if you will. And another example that actually is probably more straightforward to make the connection with the population level dynamics is the case of the calving season in wild beasts. So wild beasts, whenever they all reproduce at the same time of the year. And this is, of course, related to reproduction, and it may have important effects on the level of the population. So just to make a little bit more clear my point, by collective behaviors, I don't only understand the collective movement of individuals. I understand any kind of behavioral synchronization. And by this, I mean that synchronizing reproduction is also a collective behavior. Okay? But these two examples also provide us very good examples of cases in which this synchronization of the behaviors is not perfect. And this is actually that to me was very interesting and super cool. In the case of locusts, what we observe, or what people observe in experiments back almost in 2000, is that when you expose a population of locusts to a very high density of neighbors, so you really force them to become social, not all of them make that transition. So it doesn't matter how much time you really show them that they are in an environment that really favors sociality, Some guys remain solitary. They don't want to be involved at all in the social behavior. And with the case of calving in reproduction in wild beasts, it's pretty much the same. We have a huge peak in March, but then there are also some guys that somehow reproduce in February or in January. And of course, January is more interesting because February one may have the argument of, well, February, March, that's... But it's actually, the distribution is broad. It's not narrow, okay? So then the question that came to our mind when we started working on this project is, by observing these not perfectly synchronized collective behaviors, how the question that came to our mind is how these microscopic collective behaviors may impact the population level processes, and whether that lack of synchronization has some role in the consequences of this collective behavior, basically. So first, we want to understand how these collective behaviors emerge, how they impact the population level processes, and when understanding these collective behaviors, we cannot neglect all these features that the collective behavior has. So recapitulating the questions, first of all, we want to understand what are the causes of this self-organized process, so how individuals feel each other to really coordinate, and then how this behavior scales to much larger scales. And to do that, again, because in macroscopic systems, the scale of collective behaviors and the scale of the population level processes is very different, we work with microbes. We work with the social amoebic Dictyostelium discoideum. I'm going to show you in a while some movies of this organism and how it behaves and give you a biological background on the organism, and then I'm going to move forward to the project. So first of all, I want to introduce you all the people involved in this project. It was Fernando Rossini, who I think many of you know him. Then Allisons, who used to be a postdoc in Princeton, working with Thomas Gregor, who's a professor in Princeton at the Institut Pasteur, and Corina Tarnita, who was both my postdoc supervisor and is Fernando's PhD supervisor. So why Dictyostelium discoideum? Why we use this organism to study this specific question? Well, first of all, because it has a life cycle that is actually super interesting and shows all the features that we are really interested in. So Dicty is a unicellular organism. It's an amoeba that feeds on bacteria. And whenever they have bacteria, they are unicellular. They feed on they eat their bacteria, they divide, and they do their unicellular things. But when they run out of food, when they consume all the resources in the environment, then they trigger a multicellular phase. So all these cells, when they don't have food, they start communicating with each other, they start signaling with each other, and they form multicellular aggregates. And for those of you that don't believe me or haven't seen this before, this is what happens when you put some Dicty cells and they don't have food. But for a while, they are unicellular. They are trying to find food, and then suddenly they form this multicellular aggregate. And then once they form the aggregate of cells, they start like developing an actual multicellular structure. So some of the cells, they form fruiting bodies, basically mushrooms for physicists. And on top of the fruiting bodies, we have some cells that become spores. They go into a dormant state, they sleep, they minimize their energy needs, and they get ready to be dispersed to new environments where hopefully they will find resources.
Can we see it another time?
Yes. Once I finish this sentence. But within the fruiting body, there are some other cells that would be the stalk, and those cells die. Their only mission in the fruiting body is to actually raise the spores from the soil to allow them to have higher chances to be dispersed by, for instance, an insect. Right? For an insect that goes through the soil where these guys live, it's way easier to catch the spores if the spores are a little bit up in height. No?
Sorry, when the spore gets injected successfully, it's one cell, right?
So the spores, in the fruiting body, you have actually this kind of structure. It's like a ball of spores. and when this ball is caught by some insect and is dispersed to an environment, they germinate and they recover the unicellular phase.
Exactly. So they're vectors for the unicellular.
Yes.
So you're positive that the entity is unicellular.
Well, the life cycle is unicellular and multicellular. It has two phases. It has a unicellular phase and it has a multicellular phase.
But there are individuals that never experience multicellular phase.
If I respond to that question now, I miss 30 minutes of talk.
Do they reproduce when they are unicellular?
When they are unicellular, they consume bacteria and they divide and they reproduce.
While when they are in the multicellular phase, they don't, metabolically they are not.
No, they become dormant, the spores become dormant, and they don't reproduce. So movie again for Mateo, and for all others. So this is the movie. We have the unicellular phase, cells are moving around, and then at some point, they form the aggregation. I mean, this movie was made by Fernando in the lab, and we are interested on the aggregation process itself. So therefore, you only see cells moving, but there is actually other movies that you can find on YouTube where people actually track the concentration of the signals that these cells are secreting, and those movies are even more beautiful because they form spiral waves, and then cells track the spiral waves, and the aggregation pattern that these guys have is insane. When I arrived to start my postdoc, this sort of movies was the movies that they showed me when I had to decide what to work on, and well, I was almost as excited as Mateo, and I turned up working for 6 years on these questions. So questions up to here about the life cycle of these guys? It's summarizing. It has a unicellular phase where cells divide, and then they become multicellular, and within the fruiting body, spores are very, very resistant to the lack of nutrients, where unicellular cells are not.
So perhaps a trivial question, but you say that some become dormant and other dies. Physiologically, what do you mean? Or just that those that dies, they don't reproduce anymore?
The ones that die don't reproduce anymore. So what I mean, very roughly explained, is that if I take these cells and I put them back in a plate with bacteria, they recover the population. If those cells don't. Of course, this sacrifice, although this is not the best term to be used, but so we all understand each other, of the cells in the stock, has some fitness cost related to the formation of this multicellular fruiting body that has led to a lot of game theoretical work on trying to understand why committing to aggregation, cost of benefits of aggregation. So those of you interested on game theory, there is also some work related to this on game theory. But I'm not going to go into this today. What I'm going to, yeah, there was, sorry. Yes, sir.
Before this spore is formed, there is no differentiation, right? Even in the multicellular phase, moving multicellular.
So in the multicellular, once they form the aggregate, they form a clamp, and then they form a slack, and the slack migrates, finding like a light and has like a reverse thermotaxis. And in the slack, there starts the cell differentiation starts in the slack. There are some cells that position on the front of the slack, and ones that go to the bottom, and those will then become spores and stalks. So the whole process starts right after the aggregation process. And the whole life cycle takes about 24 hours. Okay, so there is really a cost in terms of time to form these multicellular structures. So now, going back to the movie, what I want you to pay a little bit of attention to is that although most of the cells are gone now, they all formed these multicellular groups and they left, there are some of them that actually remain in the plate as unicellular, right? You can see here that there is a concentration of single cells. Here there is some more cells that are alone. Here the slag is leaving some cells. Some of them are, of course, this is super noisy. Some of these cells are dead, other ones are not. But there are some cells that do not participate in the collective behavior. So this recovers what I was claiming in the beginning, that these kind of collective behaviors, sometimes they are not perfectly synchronized. There are some guys that do not follow, if you want to say, the trend. They don't follow the general collective behavior. And the question is, why? Why they don't make it to the group? And what are the implications that these cells have, if any, on the larger scale ecological processes of these organisms? Okay? So, yeah, sure. between the cells that don't aggregate and the ones that aggregate. We didn't do any genetics, and I don't think there is no, genetic difference. There is not. What I don't know if there is, it's not very well known is if there is any difference in expressions or whatever. But all these cells, in principle, they belong to the same strain. So far, we'll go to that back later, but we can assume they are genetically identical.
So, more evolutionary question. So, like the ancestor of, they had ancestors that were purely multicellulars, or they were unicellular and then they evolved multicellularity?
Sorry?
Huh?
So they.
What I mean is that if you look at the
They were unicellular and they evolved multicellularity. They evolved multicellularity.
They evolve multicellularity, so they don't have a purely multicellular ancestor.
No, I think. So then the first question we have to ask, what would you ask if you see this and you want to study these cells? What's the first thing that you would try to find out? What's your first question about these cells? The percentage? Okay, that's a fair question. Someone would ask something before?
Can the cells that were left behind aggregate later?
Yeah. Are they alive?
How many times they can make this aggregate?
Sorry?
During the lifetime of this unicellular, they can make the multicellular organism as many times as they want.
So in the lab, we haven't been able to do it many, many times because of mutations and things get super messy, but they can.
Separate, they can
Yes.
again.
And actually, that's the first. That was the first question I was looking for. So, are they alive? Can they recover the life cycle again, or they just die and then?
Is the distribution of nutrients exactly homogeneous and isotropic, or is there any environmental difference within the center of
So, in the plate, the distribution of nutrients is actually as homogeneous as it. I mean, the experimental took almost 1 year, as like making the distribution of nutrients super homogeneous. So I would say it is, because if not, he will come back home and he will kill me. Huh?
There is no systematic from the environment.
No. In the lab, there is no. Of course, in the environment. Actually, one of the things we want to try now is creating this more realistic environment.
They may become food.
Sorry?
They may become food.
Cells for, no, they don't eat themselves. No. They are not cannibals. So going back to the question, are they alive? Can they recover the multicellular phase? How many of you think they can? Only? If they are not alive, I'm not here. I mean. So to test that, Allison, at that time she was a postdoc at Princeton, she did an experiment that conceptually, almost as many experiments, not the one that Bill was showing, but conceptually is super easy, super simple. She took some bacteria, she put some bacteria on the plate, and she put some Dicty cells. Allow them to feed on the bacteria, consume all the resources, aggregate, and form the fruiting bodies. So this is the first day of the experiment. We have the cells with bacteria. Then we go to the second day. They already form the fruiting bodies. And then the third day, we remove the spores that we know that they will germinate and they will recover the multicellular phase. And we give, or she gives food back them. She gives them food back. So she puts more bacteria on the plate. And what happens? What happens is that these cells that had an aggregate, they start dividing and they consume the new bacteria and they form the multicellular phase. They form the aggregates again. So regardless of the particular mechanism by which these cells become aggregators or non-aggregators, I'm going to go to this in a while, these cells are alive and they divide and they can do, let's say, so by now, something.
You're saying that there's an energetic threshold that they might get in order to go back from their multicellular phase, something like that.
So when they, the question is if I understood correctly, if there is an energetic threshold in the state of the cell that determines the fate of the cell. For sure, the energetic state is going to matter, but all the experiments and all the models we did assume that they are in the same identical energetic state, and both our theory and the experiments agree very well. I'm not saying that it's not one of the variables, but our Our suggestion is that, what we suggest is that it is not the main driver of this population partitioning process. But I will go back to this like in 3 slides. Okay, so we have these cells. Some of them, they aggregate, some of them don't aggregate, and the ones that don't aggregate also are able to reproduce. So the point is, of course, that the whole point of aggregating is to resist the lack of nutrients. They become dormant and they live longer. But as I was saying before, when they become dormant, they also need some time to form all this fruiting body. There are some cells that die in the stalk. So it's not so trivial to say, I don't have food, I go to the multicellular state. Why? Sorry? Loud, don't be shy.
You can die.
If you go to the multicellular state, you can die. But if you don't go, maybe you in principle, you die as well, no?
Yeah, but this is like in ants, when the workers don't reproduce but help the queen to reproduce because they are genetically very close, maybe?
That's one possibility. Do we have another possibility? No?
They hope that more food comes from the heaven.
That's a very good point. If I commit to a process that takes me a whole day, maybe someone is feeding me during that day and I'm missing the food. So if the food is going to come back very quickly, I cannot guess. But if food comes back very quickly to the environment, then I don't want to invest energy, time, take the risk of dying. I'd rather stay alone and wait for food to come back, right? So there is a kind of trade-off between deciding. I'm going to anthropogenize a lot these cells, but there is a trade-off between becoming part of the multicellular structure and staying solitary.
We think about getting over.
Yeah. So then, to move on these questions, we constructed a theoretical model to explore. Yes?
Maybe, did you guys measure like that rate, something like that, so that you consider cells dying after going to the multicellular state?
Cells that don't become they don't go to the multicellular state?
Did you guys measure like that rate?
So there is, for cells that don't become in the multicellular phase, you can build the survival probability of a cell as a function of time, and it looks like this. And after about 240 hours, no loner cells survive, okay? So there is a time threshold after which everyone that didn't commit to the aggregate is not alive. This is not our data. This is data by a group in France, but working on these same strains. So then, going back to Vitor's point, the question is, what is the proportion of cells that becomes aggregated and non-aggregated? So we constructed a model to explore the potential role of these cells, and we assume that there is indeed a proportion. We don't know so far. The only thing we know experimentally is that they are alive. We don't know anything else. We don't know if it's just noise, if it's a proportion. We don't know anything. We know that they are alive. But because we have some mathematicians in the group, in the team, let's make a hypothesis. And the first hypothesis is that upon aggregation, a proportion of the cells aggregate and a proportion of the cells don't aggregate. And going back to your question, cells that don't aggregate, they die way faster. The other ones die way slower. And we assume also that this aggregation partitioning behavior is heritable. What I mean by this is that in subsequent processes of starvation and aggregation, this alpha trait is fixed. So I can identify different genetic variants of this organism by the value of its alpha. Okay, so I will have, instead of red, blue, green, and orange cells, I will have alpha 0, 0.1, 0.2, 0.3, whatever. I discretize the trait, and I run my model. And what we find is that if we label these genetic variants with different values of this trait, and we allow them, a bunch of them, we take, let's say, 50 genotypes, we discretize the interval between 0 and 1 in 50 values, and we allow all them to compete in environments that are labeled by the time between the consumption of the resources and the arrival of the new pools of resources. So bacteria dictate all the bacteria, and then after 200 hours, new bacteria arrive, 200 hours, new bacteria arrive, and so on and so forth. Okay? So when we assume this, we find that if the time between the consumption of the resources and the arrival of the new one is very long, which one would you expect is the optimal alpha? What's the best thing I can do if it takes 3 years for food to arrive? Alpha 1, right? Every cell should aggregate. On the other extreme, alpha equal to 0. But now let's add a little bit more of complexity. What happens if these starvation times that I was mentioning before keep this mean value, but they fluctuate? Now the question is not so trivial, because sometimes I may have a starvation time that is super long, so then it makes sense for all the cells to aggregate, but then I have one that is super short, so then it makes sense not to aggregate. So what we find is that, as Mateo is suggesting, over the continuous of different environments that are labeled by different mean starvation times, these times between the time at which you consume all the resources and the arrival of the new one, they are random. They follow some distribution. But as a function of the mean value, we find that actually the optimal value of alpha is a continuous function with the environment. So the optimal aggregating behavior is indeed to say, well, some cells, I will put some X on this and some X on this other side, so then I minimize risk, and on geometric mean, I maximize my reproduction success. Okay? And actually, this is a very interesting result, because if we now couple this result to some environmental heterogeneity, so these environments will allow dispersal of spores between them, then what we find is that as the dispersal increases, the diversity of strains increases as well. So because of this partitioning between aggregating and non-aggregating, we are increasing the diversity of these species out there. So we allow for some values of the dispersal, we even have almost all the different strains coexisting. And of course, eventually, if we continue to increase dispersal, we somehow recover a mean field limit in which, on average, all the environments are identical, and then I only have one strain. So for intermediate values of the dispersal, diversity of strains is boosted and is very weak, both without dispersal and a very high dispersal. And the same happens if I consider a different type of environmental heterogeneity, which would be seasonality. So for some time of the year, my environment is super good in terms of short starvation times. And for other period of the year is super bad in terms of super long starvation times. So I have a wet and a dry season. In that case, we are also able to have stable coexistence of species oscillating through time. The process by which these species coexist, for those with more ecological motivation, is the storage effect. The storage effect basically means that during one season, we create a storage of one of the strains, and then when the environment switches, that strain declines, but still remains, and the other one grows, and then that leads to stable oscillations with a very hand-wavy explanation. So,
when you do the calculation of changing the mean time between that you wait and starvation, if the distribution of times had a long tail, would it matter?
So the shape of the distribution in the when you don't have seasonality, what matters is if you have a fat tail, it matters. But even if you don't have a fat tail, is the variance of the distribution what matters. So you need enough environmental variation. When you have seasonality, if you have fat tails, you lose the coexistence. You cannot have long fat tails because you indeed need to have well-defined seasons if you want. So even without fat tails, if you have, instead of these Gaussians that have almost no overlap, if you have exponentials with very different means, you don't get it. Because what is important is the overlap between distributions in order to be able to create this storage. Otherwise, you don't have it, basically. So this is a super computational model because it has a lot of ingredients. Something we are trying to work on now is to create a real toil model with all these ingredients to gain analytical insights on these questions. For instance, what are the properties that the environment needs to meet for having coexistence? What types of fluctuations allow me to have coexistence? And these sort of questions. Because this is, as Mateo was pointing out before, when you have environmental fluctuations, you expect the evolution of these bed hedging strategies, right? I don't have all my eggs on one side or on the other, I kind of split. But what we are showing here is that when you have two different sources of environmental heterogeneity, indeed what you have is, you may have the evolution of a new bed hedging strategy that outcompetes any other bed hedging strategy, but in some conditions, you have coexistence of two different bed hedging strategies. So which are the environmental conditions that determine one outcome versus the other is something that we are trying to figure out with more simplified models to be able to gain analytical insight. But going back to the question today, yes?
Are these bed hedging strategies, they're consistent within the lineage? Like, it doesn't change, maintain the...
Is that your question? So that's a super, super important question. The question is, if these bed hedging strategies are conserved for a given strain through the whole life, if you want. So are they meaningful, or what's going on here? Because I said I was assuming that this is a heritable trait. I was assuming it. I didn't have any experimental evidence on that. So then we need this experimental evidence, and to get that experimental evidence, we were very lucky that Fernando spent a long, long time working out these experiments. And what he did was to count the number of cells that don't aggregate in the plate as a function of the number of cells that he's putting on the plate. So basically, he puts different densities of cells in the plate, allow them to aggregate, and then counts the cells that didn't join the multicellular phase, simplifying 4 years of his life. So what did he find? Super interesting results. First of all, when the density of cells is very low, cells don't aggregate, and then every single cell in the plate is a loner, a non-social, a non-aggregated cell. I'm going to call them loners to go faster. This is known. Aggregation is triggered by density, so then if I don't have enough cells, they don't trigger the collective behavior. Nothing new here. But now what would you expect? If I increase the density, what do you think is going to happen? If I have a proportion of cells that don't aggregate, then I should expect something like this, right? This would be alpha. If every time I put cells, 20% of the cells don't aggregate, then as I increase, I have 20% of the initial population. How many of you think we got this? I wouldn't be here if we got this. What we got actually is that the number of cells that do not become part of the multicellular phase after some threshold is independent of the initial cell density. So it doesn't matter how many cells I put in the plate, there is a threshold in the density of cells after which they don't aggregate. And as you can see for this particular strain, the variance in this trait is super low. It's like a well-defined quantity with a narrow distribution. And we can repeat this experiment for different strains. And with a little bit more of noise and doing more statistics, they leave behind different densities of cells. So what's the take-home message here is that, yes, the trait, the partitioning behavior is conserved through the life of different strains, and that different strains have different partitioning behaviors. But it's not the proportion. So all the results I showed you before fail because they have assumptions that are not correct. So then, what are the causes of these loners and the ecological consequences, considering that we measure this new we have this empirical result that now breaks down our previous hypothesis. And yes, I thought you were going to stop me.
No, I wanted to ask a question. How many aggregates do you form as a function of the number of cells you plate? So, in other ways, how big are the aggregates as a function of the cell densities?
So they decay. The more cells you put, the few so the aggregation territory, the size, decays with the initial density of cells.
Okay.
So the typical size.
So you tend to have a typical size that is conserved independently of the density of cells.
Sorry again?
The typical number of cells that go in the same aggregate,
Yes.
aggregate is constant.
Yeah, we think it's size regulated. So the question, what are the causes of these loners? What we propose, again, we haven't measured expression during aggregation, or we haven't measured molecular paths or anything. We propose a mechanism. That is that the origin of these cells is the result of this decision that is stochastic and depends on the concentration of neighbors around you. What I mean with this, with pictures, what I mean is that initially my cells are all without food, they are all signaling with each other, I'm super crowded, so the concentration of signal is homogeneous in the space. And they are all above this threshold that I was showing before, okay? So I'm not in this region with I'm in the region with a flat result. And then some cells are measuring the environment, and some of them, with some probability, they start the aggregation process. Those ones that are the ones that are yellow here start moving towards the aggregation center, and as the aggregation process starts, then we have a spatial heterogeneities in the concentration of signal. In the way that some cells that here are brown, that before had the chance to join the aggregate because the signal was super high, now they can't, because now the concentration is low. And then as the aggregation continues and continues, at the end, we have some cells that got stuck, basically, in the unicellular phase because they made the decision to aggregate super late. Once they wanted to go, to take the bus had already gone. Now they have to wait for the Uber. So, of course, this is an individual-based model. I'm going to show you the analytical limit now, so you get more insight. But what is important here is that, of course, the results, the number of loners, will depend on the parameters of the model. Here we have different parameters. We have the velocity at which cells move. We have the intensity with which they signal, the sensitivity to that signal, the probability of making the transition. We have these 4 parameters that are basically key. We have the diffusivity, which is an environmental variable. But what is really, really important is that regardless of all these parameter details that will give me a quantitative agreement, this model is able to recapitulate the 2 main features of this macroscopic pattern in terms of the collective behavior that we were showing before. So if my initial density of cells is very low, of course, aggregation doesn't start because I'm imposing that in the model. It's not surprising. But I'm not also interested on that. But as I go above the threshold, then I get a plateau in the number of loners that is actually my experimental result. Something to give you the whole picture. What we cannot recover with this model is the variability that I was showing before in data. Variability is not, it's something we are working on. So this is mean number of loners over several simulations of the model. So I run my model 1,000 times, I count 1,000 times the number of loners, and this is what I get, the squares. Questions up to here?
So the loners are not genetically different. They're all
Yeah. No. Okay, so of course this is computational. Once we had this, biologists were super happy because we were explaining the experimental results. But as I was saying before, we have some mathematicians, one physicist, and we wanted to really understand whether this proposal of this lack of synchrony between the 2 different scales, we have the scale at which they move and the scale at which they decide, whether that's the key ingredient for our model. And then to do that, you can recover the mean field limit of our model. So basically, now I assume that the signal is everywhere. Whenever I send signal, it diffuses perfectly and is homogeneous. So I make my decision in terms of the number of guys that I have in the system with an infinite neighborhood. So basically, when cells are pre-aggregating, they are in this blue state that we were discussing before. They have a transition rate to an aggregating state. And that transition state depends on the same population of cells. It's a density-dependent transition rate. And then they do a second transition from aggregating to actually being aggregated. That depends on the velocity that they have and a characteristic spatial scale in the system. So we have 2 transition rates, but the important thing is that to jump from blue to yellow, you need a critical size of the population. So at some point, as transition from blue to yellow happens, it will stop because the transition rate will become 0. And we can write that in a set of 2 ODEs that looks very, very simple. But remember that this lambda is not so simple. It's kind of a heavy-side function for those with more mathematical insights. But anyway, we can work out what happens when the population size is very low, is very high. We can do some mathematics on this, and we can calculate the value of these P cells when n tends to infinity. And what we find is that actually depending on the ratio between the 2 transition rates, this plateau is either finite or is equal to 0. So the plateau that we were finding computationally is also recovered in this analytical limit without any space, just because of a matter of this lack of synchrony in the transitions. So we have a kind of a phase transition, if you want, from one result to the other. So now I'm going to go a little bit quickly because I lost kind of the sense of the time. But the important thing is that with this result, we can take a bunch of our strains and we can rank them according to their aggregation performance. So I can define a one-dimensional space that I call aggregation performance, and I will place these strains in order depending on how well they aggregate. So the orange one leaves fewer loners and is the best one, and then the green is the worst, and I can rank them according to this single trait. And then going up to the ecological questions, I can ask what are the ecological consequences of this particular behavior that I found. And this is, for DICT, is super important because DICT is super diverse. If you go to the field and you take a piece of soil like this, you find like an incredible diversity of species. It's super diverse. You have a high coexistence. So how is that coexistence maintained? It's a very important ecological question for this organism. So actually, when you have these 2 different strains and you mix them together, they don't separate. So if you put red cells and blue cells together, they form this kind of purple fruiting body. So question here. How are these different aggregating behaviors interfering with each other when they are together, no? What do I find? I find linear combinations. I find one dominating, the other one dominating. So if you do that experiment, what we find is a weak effect, but significant. I got this question every time I gave this talk. We get that there is actually interactions between interferences between the aggregation processes that make the behavior of the sum of both strains to be different from the behavior of the combination of both strains to be different from the sum of the behaviors, if you want. So there are no linearities in the aggregation behavior. This is experimental data. Our model recovers not only the experimental behavior, but through the parameter space, a bunch different types of interactions. And of course, one of them is the experimental one. So why is important the model at this point? I mean, I've been talking about the dialogue between experiments and theory. And the reason why the model is important here is because so far, when we do mixes of cells, we cannot distinguish the identity of the cell. So when I mix red and blue cells, I cannot say how many cells are red and how many cells are blue at the end. I can do it in the beginning because I know, but at the end we cannot. There are some ways of doing that, but we are working with wild type strains here, and you cannot do the sort of fluorescence that people can do in other types of strains. So the model helps us in understanding what's going on, or at least proposing which could be the identity of the cells that we are finding in the experiments. Of course, this is all exploration because the model is well calibrated for the single strain case, but who knows if it's failing down here. So what we find is actually that as we mix the population in different proportions, one of the strains, in this case that reminds the experimental case, one of the strains goes very quickly to 0 in the number of loners. So somehow the presence of one of the strain is making the other one to be super good and to aggregate perfectly. It's dragging, one of the cells are dragging the other ones. Intuition behind them, well, if it aggregates very bad, then those cells stay there and help the cells that got stuck to actually move away. No? There are some cells that are left behind that keep the quorum for aggregation to happen. But if we map these strains in our map, in our space, what is actually happening is that because of these interactions, strains are behaving more differently. Why? Because the one that was leaving few loners behind now doesn't leave any loners. So it's actually moving towards the extreme of the aggregation performance. And the other one that was leaving some loners now is leaving more and is moving to the worst extreme. So strains are actually diverging in their behavior, in the way they behave. Questions? Everyone lost at this point?
Is it a case of specialization?
What do you mean by specialization?
For instance, again with ants, some behave in some particular way and the others in other.
So we only do one aggregation process, so we cannot track if one cell always is left.
As a social group, one social group becomes more efficient in one task and the other on the other, and the whole population is better in this case.
So that's, that we don't know. I mean, we don't know if they, if I understood the question correctly, if we, you mean if as we repeat this experiment many times, if we observe a move towards better performance in aggregation, no? In one of the populations, for instance. That we cannot tell because we cannot do this experiment many times. As you repeat these 3-4 times, they mutate, they start mutating, and everything becomes super noisy, and we don't have data on that. But we try to do it. But so, who is a biologist here? What may happen if I have, let's say, an island with 2 species, and they evolve to behave more differently or to occupy niches that are more different? Sorry? When they behave more different, they can get to, I mean, it's something that remains to character displacement, right? You have 2 guys that have, let's say, they need the same food in the beginning, but through different generations they evolve, and they need more different food. And that could help coexistence of the species, right? So imagine that we all here like picanha. But then a group evolves to become vegetarian. Then there is less competition for the meat, right? Because one's with food, one's with the other food, and then we may coexist in the environment. So then the question that we ask here is whether that's happening because of these shifts in the behavior, if this can help the coexistence of a species.
Can I ask one more thing about the experiment?
Yes.
So when you put them together, you always see that the orange and the green, they all aggregate orange with orange and green with green.
Well, we mix green and orange. We mix green and orange on a tube, we shake them, and we plate it, the mix. And then they aggregate together, and we count the number of loners.
They aggregate together?
Yes.
Okay.
They don't, I mean, they have some weak discrimination of identity, but they form groups that are genetically heterogeneous. Yes?
I've seen some, colony studies in some species where there is an aggregation rate. There's also a disaggregation rate because sometimes it fails and so forth. So do you guys see something like once an aggregation starts, does it have, by any chance, like disaggregation also might happen sometime in the near future?
So the question is if we observe aggregations that start and somehow are stopped. What we observe is some cells, especially the ones joining later the aggregation, that they become solitary around the group. But we don't observe a completely, an aggregation that is completely shut down. So explaining very quickly what we did to test this hypothesis of this divergence of behaviors leading to higher diversity of strains. We took these strains, orange and green, if you want. They are labeled in our model by different parameters. And we allow them to aggregate and reproduce through different periods in our model. So instead of aggregating them once, we allow them to aggregate many, many times. And we do that in 2 different conditions. In the first one, we force them not to mix, as Fabio was asking. They distinguish each other perfectly, and they segregate. So we don't have these interactions, basically. And what we observe is that there is some competition for some time, and at the end, every different environment, these environments, each square is an environment, with a fixed starvation time as in the beginning, and there is no dispersal. They are independent, different islands. I have 25 different islands with the same environmental conditions, different initial conditions, and they compete for a long time. Everyone is green at the end. If now I allow them to mix, what I observe is, first of all, the time to extinction is shorter. Why? Because they are more different. So the environment is selecting faster for the one that is better adapted. And the second thing that I observe is that different environments select, at the end I have different winners in different environments. Why? No intuition? I hope there is not. It took me a month or 2 months to... Well, actually, because the extinction times are so short here that environmental fluctuations allow to keep the environment for a while in conditions in which the orange is able to win, to outcompete the green. So indeed, this environmental, the general idea, the general point, is that these shifts in the behavior, together with environmental fluctuations, increase the diversity in at different scales, if you want. In this case, I have lower alpha diversity. My coexistence times are shorter. So in principle, if I look at one patch, I have lower diversity. But if I look around patches, across patches, then my diversity is higher. So I have higher beta diversity for ecologists. For physicists, species are less time in the same place, but they are in different places. Yes?
I got a little lost on the first one. The orange ones are not so good consumers of the resource, or what is it?
So imagine I have you remember this? This line that was for the other model, but doesn't matter. We remember this problem? We take environments, of course, here in the middle, because they are the funny ones, the other ones. And then the orange and the green compete in one of those environments. And we say, okay, I put 50-50. Let's say mines are white and black now. This is the first generation. In the second generation, the whites are a little bit higher, and eventually they are all over the place for a very long time. But when we allow them to interact from the very beginning, then this time is shorter, but I have a probability P of black and a probability 1 minus P of white. So there is uncertainty in the winner of the competitor.
So it's not like in the second one, the orange will always die.
No.
But the only difference is that you mix
The only difference is that they interact. And by interacting, I'm dragging your behavior towards mine or further from mine.
So when they're segregated, like you put the two populations and split the
Mathematically, in the model, I force them not to interact. No, this is theory. This is not experiments. Sorry, we took the model and ran it several generations. I thought I had said that. I said we cannot run this experiment many times. So this is, with the model, we test possible implications of these shifts in the behavior. Again, even the shifts in the behavior, we cannot track the identity of the cells. The shifts of in the behavior are model suggestions that agree at in some limit with the experimental result. But, and this is very important, sorry that I go back. This is all the information we have from the experiments. Here I don't know how many cells are red. Well, here I do. In the other, I don't. Something we are trying to do, but it's not easy. So then that's why I take the model and I say, okay, in this curve that looks like the experiment, what is the identity of the cells, what's going on, and what could happen? Why? Because these experiments are not easy. You have to convince someone to, again, spend 4 years of his or her life doing this. They are not cheap. And it's good to have some intuition of whether it's worth. And that's where the model is helping to decide whether it's a good idea to pursue these experiments or not. So again, the idea is that this shift in the behavior change diversity at this scale, in this case. If you mix different pairs in the model, you find different shifts on the behavior and different changes in the diversity of the species. But I'm not going to bore you with this. I see already many tired faces. I'm not going to say anything today. Today's fault of the speaker, it's not yours. And well, beyond these ecological implications that I wanted to focus a little bit more because of the focus of the school, all these questions have very interesting implications beyond that. So for instance, this issue of being part of the stock or being part of the spores, what's the role of these cells that don't aggregate, that maybe later aggregate and help recover in this multicellular phase? There is many questions that go beyond ecology here. But again, I'm not going to go to that. I just want to finish with 2 slides. I promise it's only 2, where I already introduced some of the things we do here in the beginning, but I kept some of them to the end. So in case any of you is interested on them, we can talk. So this is maybe more important because it's future. This is past. So one of the things we are doing is we're using these models of population dynamics. For instance, these Lotka-Volterra models that usually assume that all individuals die at a constant rate. We are studying what happens if these individuals become old in different ways, how the equations change and whether this changes the patterns of exclusion and things like that. This is something actually Jairo is doing. He will finish by next week, I hope. No, I'm kidding. And maybe the main thing that we are trying to do now is to study what happens to these population level processes when there is a spatial structure in the population behind. And we are doing that on different systems. We are working with microbiologists to study pattern formation in bacteria colonies. This is an experimental picture of some bacteria. And this is the model. This is what we are doing with microbiologists in the US. And on the very large scale, we are working with large-scale patterns of vegetation, so how plants self-organize in the space and what are the implications for ecosystem-level processes. All this is using statistical mechanics spatial models and reaction diffusion equations. Hopefully, Gabriel, maybe it's interested in working on it. The picture is a little bit. And of course, we are always looking to new systems and new biological systems to explore these questions of pattern formation. 2 of them that we are thinking of is the clustering of bugs. This is a picture I actually took in my hometown. And mussel beds, there is many groups working on that. So at these different scales, how these individuals form these spatial patterns and how that changes processes at the ecosystem level. With this, I finish because always the one that is pushing people to be on time is, but I had a lot of questions, so it's not only my fault. Well, I want to make it clear again that we are looking for postdocs, PhD students, master students. We have fellowships, so that's something that maybe helps. You have to apply for money. This is my email if you want, but I will be around the whole week. Well, the references of the papers and always people that pays that, well, allows us to eat. And thank you very much.
Questions?
Thanks. In the hybrid fructiferous body, in the hybrid, in the mixed, what is the identity of these spores? Are mixed also, or pertain for only one strain?
So we know that. So the fair answer is we don't know because we don't label them, right? We don't label them. We don't know. But there are many experiments that actually label these cells, and they are mixed in the spores. They form, I mean, the body of spores is mixed, but this is very important. The proportion of let's say, red and orange in the spores is not the same as the proportion of green and orange in the initial population. So if you start 50-50 and you count the spores, it is not 50-50. And that's what has led to many studies using game theory to say, well, there are some cheaters that are more present in the spores. But yeah, people that count the spores, many times they don't count the stock and they don't count the cells that don't aggregate. So if you have this trade-off and the cells that is more present, more frequent in the spores is less frequent on the other one, then this whole socio-biological thing needs to be taken carefully.
Okay, thank you for your talk, Ricardo. It was very nice.
Thank you for staying.
In the future prospects, are you thinking in, with these aggregates or lone cells, how is the top pressure or predator pressure to see how it happens in natural systems? Do you consider that maybe a proportion of aggregates and lone cells could be related with that also, or signals from predators?
You mean like considering the role of predators on these cells?
The role of predators, the pressure on aggregates or lone cells?
We haven't thought of that, but I mean, what is known is that the formation of these aggregates is a defensive strategy, right, against predators. They become bigger and they cannot be eaten. So that's a very interesting question. We haven't explored anything.
Thank you. Did somebody in your group try to analyze this with game theory, and in particular with kind of multi-level selection or group selection? I mean, I know that Corina is a fan.
Can you switch off the... No. Short question. Sorry, I was joking. His question was if we've tried to use group selection or kin selection to study this. No, our hypothesis is that it's really considering the whole picture and really considering the ecology and the context in which these behaviors evolve, what is more meaningful to explore these behaviors.
Other questions? Okay.
I'm still curious about your mixing and not mixing situation. Mm-hmm. How it makes a big difference on those experiments? You have lots of resources, scarce resources. Are they uniformly distributed on the plates? Can you elaborate a little bit more on that? And also, can you quantify how mixing, how well mixed versus no mixed?
So one by one, the questions, the resources, as I was saying before, are homogeneous.
But a lot of resource or not enough resource?
So you put the resources, cells grow, you remove cells, you wash cells, and you put them and they aggregate, homogeneously distributed.
But you're not starving the cells, right?
Cells are starved in the washing. The cells starve, then they are washed, and then they are played to aggregate. They don't starve and aggregate. So we don't have, because that's an important question. If you put cells to feed on the plate, consume the resources, and aggregate, and you want to count these cells, for the first experiment, it doesn't matter because it's a qualitative experiment. If you really want to quantify, to make numbers, when you plate cells, they can be in different physiological states. One of them may consume resources faster, you may have regions with higher density, and they may consume resources heterogeneously. And although your resources are homogeneous, they can consume them heterogeneously, and then that biases your results. So for counting the actual loners, they are washed and they are played without resources in an homogeneous way. And the agar, they are put on some agar, and the agar is, we actually explore also the role of the concentration of agar, and it's also an important variable. So the answer is, when we count loners, they don't, they are put without resources.
So, but that makes the experiment, the size of the plates very important because you have a mixing time versus a starving time, right? That has to decide on the size of your plate.
So you mix cells, you put a drop of cells, and they aggregate in the size of the drop, they aggregate. What is important is the concentration of the agar, for example, because it, among many, many other things, it controls the diffusion of the molecules. And then you have different sizes of the aggregation territory. You have different levels of loners, for instance, because you are somehow controlling the environmental conditions, if you want. You are controlling diffusivity with this parameter. But when you put a drop of cells, they don't have any range expansion or because they don't have food. They just start aggregating.
I'm just curious because in the end, you present your new work on pattern formation on bacteria. And on some old papers we did with Ashley Jacob, on this sort of finger growths on bacteria, then you get nice patterns when you start with bacteria. If you have a lot of food, the patterns are kind of boring, right? So
well, so actually in this, I mean, I felt I was being super long, but in this experiment, we indeed have a microfluidic chamber. We put few bacteria, we seed bacteria, and we give them a lot of food forever, and they grow. It's a range expansion. So in that sense, these experiments have nothing to do with dicty. Actually, these cells are genetically modified, so they don't move. They are like sessile guys that divide. What we play with is with the adhesiveness of the cells.
Hello.
Hi.
I'm really shy, but I will try to make a question. You talked about the strategies and decisions, and I would like to know if you think it's possible to approach this kind of patterns and systems with game theory.
So as I was saying, this problem of multicellularity by aggregation, the formation of the fruiting bodies, there is a lot of work using game theory. They are trying to say, what's the benefit of being part of the group? What's the cost? There are some cheaters in the context of game theory, some guys that are always super present in the spores and they are not part of the stock. So there is work using game theory trying to explore this question. Our proposal is that these models are somehow limited because they say, all my fitness is determined by being a spore. And we say, well, depends on the environment where you live. Being a spore may be super bad. So what we propose is that those models need somehow to be expanded to consider the environment, to consider all the contributions to fitness, and many other variables. Yes?
So first, thanks for your talk. It was very nice.
Thank you first.
So I was just wondering about like some species distribution like affects the probability of one individual relating to its surroundings, right? So does it have like a known distribution of these beings there? Is it random?
You
like
mean
they're randomly distributed in the plate or something, or do they have like a pattern?
Sorry, do you mean in the indicti or the?
Like I mean the plate.
So this part. Yeah. So if in the plate, in the mixes, in the experiment with mixes, we mix them well in the tube and then we put a well-mixed population, if that's the question.
Do they have like a established distribution pattern, or is it just random?
It's random. So we mix them and we put them all over the place. So in general, the initial distribution of cells is random, well-mixed with red and blue. So we don't put red and blue. We mix them together and we allow them to co-aggregate.
Any other question?
Sorry, what I say we related to experiments is Fernando.
Any other question? Okay, then let's thank Ricardo again. And so we'll see over there